Good afternoon, everyone. This is Justin Howard bringing the second and final presentation to you in our Biology 503 class. My plan is to go through this presentation concisely, hopefully make it a little more entertaining and bearable for you to watch, and still maintain some professionalism while I'm doing it. What I'm talking to you about today is conservation of magnetite biomineralization genes in all domains of life and how that is an implication for magnetic sensing. And so what I want to, or the topics I want to cover are the background behind geomagnetic sensing. And I want to do the same thing with navigation because there's a few different ways that organisms could be using a geomagnetic signal to navigate or to orientate themselves. I want to talk about the naturally occurring mineral that is responsible for it. Make sure we discern some big questions that the authors are asking, and then talk about the results and conclusion, which was spelled wrong there. I just fixed that. I think the authors had some definitive results here, and so I'm going to try to back that up. In talking about geomagnetic navigation, lots of animals can do it, and some of the common ones are seals, sea turtles, bees, sharks, birds, bird dogs, I'm sure, and even dogs themselves. But there's a couple different proposed methods on how they do that. This study was presented to us by its author in our seminar class a few months ago. They were trying to discern how weddell seals navigated underneath the ice as they swam in search of food. Or they ruled out being able to use landmarks or sensing where other seals had swam because of little swirls in the water. And then also they were trying to rule out a geomagnetic map that they were using. And they had a map here in their paper. You can see where there were different intensities of this geomagnetic signal. The yellow and orange areas correspond to more intensity. Ultimately, they discerned that the seals were following the roads that the researchers made as they were going to each site where they were capturing and releasing seals, and they were able to see them from underneath the ice and follow those. So it was a landmark thing that they were using. This intensity of the Earth's magnetic field changes over time, but another representation of the magnetic field that we can discern is inclination. And so this is a computer-generated picture that shows the fields coming out of the top and bottom of the Earth. This middle here is all generated from Earth's core, but these blue arrows or these blue lines would indicate that the magnetic field is pointing upwards, like a sort of like a dipole moment, and the orange ones would be pointing downwards. And as you get near the equator, they would have to flip, depending on which way he was going. Another example of that is right here. You can see if I'm a bird and I am migrating from South America to North America, I was able to sense the geomagnetic inclination, not the intensity. Now we're talking about the inclination. That magnetism direction would flip as I reach the equator and start pointing upwards. And maybe they have like a horizon that they can sense or see in terms of this flip here that allows them to fly north versus south. I really got into a rabbit hole of being able to navigate with a compass and what inclination and declination means and true north and azimuths and things, but I quickly realized I was way outside of my wheelhouse. So moving on here, I want to just clarify what it means to sense something. Vision is one example. I've got a sensory organ, which is my eyeball. Light will enter the lens. It will be diffracted through the liquid that's inside. It will land on the retina. Rods and cones will be excited depending on what wavelength there is. You know, humans can only perceive a small portion of the light spectrum, the visible part. We've got things like infrared that's way above there, and then ultraviolet and blue light, that's shorter wavelengths. But because of that, because of these organs and those cells, I can sense depth, I can sense distance and color. Another example would be hearing. We've got not only anatomy that helps us with that, direct sound waves into our ear because of the auricle here, and then a tunnel that takes it down to a membrane that will vibrate, and then that vibration is turned into a signal usable with specialized cells that travels down a cranial nerve into our brain. In terms of the paper subject, it's been documented and it's still observable today that there's certain bacteria that have a geomagnetic sense. Here you have a picture, which I have not cited, a picture of a bacteria with all these lipid-bound organelles, these little vesicles that contain magnetite. The second hypothesis on how animals are sensing geomagnetic signals is through this light-dependent reaction. To sum it up, if you can remember orbital theory and electron spin, as electrons are jumping from a higher energy state to a lower one, light is emitted. And so that can be manipulated by UV light. And if that UV light was entering, say, the eyes of a fly or a bee, it will change the spin pair of those electrons, and there's a lot of quantum theory involved in that. Probably important to note that this study, I've quoted this from here, it was based on substantiating that the abdomen in the honeybee is where the sensing was arising from, but they've been shown to do both light dependent and the bacterial sensing reactions. It's not decided. This has not been proven yet. It could go either way, but if you had to think about it, which way is more plausible? There's argument, which I kind of fall into the camp of, that the bacterial method is more plausible because it can be traced all the way back into our ancient lineage, our ancestral kinship with those bacteria, and it's in eukaryotes today. And so this could be just a variation of that, or a newer trait. Rounding out the third hypothetical method, or additional method in which animals may be navigating with the geomagnetic sense, is found in sharks, in this organ called ampullae of Lorenzini. You can see in this picture here, they've got this shark has this pattern of pores on its ventral side, and so there's all kinds of cool pictures of what this system looks like as it's dissected, but that could be just another way that this is happening. Sharks have a great sense of sensing electromagnetic fields within the water, and there's something to be said about that, I believe. So let's get to magnetite. This is a naturally occurring, naturally magnetic ore. It could be here, or it could have formed whenever the Earth was in its earlier stages or arrived by asteroid. But if you think about snowflakes and ice crystals, how they form perfect lattices and different formations, it's important how this forms within an organism, or how it's generated biogenically. Here's a picture of some fossilized magnetite that these researchers believe came from an animal. And I just thought it was really neat how they were shaped like tiny arrowheads. You can get different shapes of crystals with this mineral, but this is just more for demonstration purposes. And so finally, reaching the big question that the authors are asking, within this trout that they studied, are there olfactory cells, which is our sense of smell? We have an olfactory nerve and an olfactory sense. Within those cells that produce magnetite, can we differentiate them? Can we separate them from other olfactory cells that do not produce magnetite? And can we discern or sequence those cells and their genetic information to see if any of those genes will show up in a blast program or sequencing? They took the trout and dissected this portion of the trout head and took out this organ, which is new to me. They call it an olfactory rosette, and rosette is just an anatomical term that I learned about during this. Took this section of the rosette, this lamina here, discerned if there was magnetite-producing olfactory cells there, and then gathered other olfactory cells from a different location. And this blue arrow here indicates the olfactory nerve, or the bulb, traveling back up to this tiny trout brain. Remember, we're looking for cells that produce it, that's biogenically producing magnetite. It's not a bacteria that's causing it. It's not forming because of pollution or some kind of additive in the water. It needs to be there naturally. And they took those cells and looked at the magnetite inside and did a bunch of different measurements on it. Some of them were new to me: atomic force microscopy, magnetic force microscopy. It's important to note that this measurement that they took here in this figure 1b, this signal is different from the magnetite you would see in bacteria that can be taken out today, which helps support their evidence that they are looking at magnetite that was biogenically produced. I really tried to discern what this nomenclature is right here, this HOE, but it was a little bit more than what I could figure out, and so I'm sorry I can't give you more information on what's happening here. But if you'll read the caption for this figure, it'll say that this sharp edge here, this decline, is what I'm thinking they're talking about, means there was a internal magnetization or an internal field developed by the magnetite itself as it was being excited by the signal that their machine was producing. Some of the tissue that they took out of that trout and the clusters contain the mineral. D here is just showing the dimensions of these clusters, how tall and wide they would be. And we're talking about a small scale here. And they also made an interesting observation on the bottom of page 2, which I will put up on the screen for you here. Their pictures and their retrieved tissue samples containing magnetite look just like some from a study that was completed in, let's see, 2000. I highlighted it here on this slide. So the authors reproduced this type of study. I believe this was done with the ferromagnetic spectroscopy. It's placed on a film, and they would insert a metal probe that would change the magnetic field, causing this light and dark aspects of the same picture, as in their current study here and the one study they referenced pictured here. These arrows indicate the direction of the magnetic field. And so I just want to stop here and say, here they've confirmed that these trout produce biogenic magnetite based on the structure of this particular crystal molecule they found within these cells. They can say, okay, this originated biogenically here and not from pollution or not from bacteria, because they all form different ways depending on their sources. Now, on to the sequencing part. The way they differentiated these cells was put the samples from the trout into a test tube along with other samples like from blood or muscle. And as they were suspended within a solution, they would put a magnetic probe down the top of the tube, and so cells with magnetite would stay towards the top and the others would sink down into a pellet in the bottom. They were able to separate them that way. Once that was done, they could sequence for RNA transcripts within the cells themselves and take those and compare them. And what they came up with was RNA transcripts that belong to approximately, or not approximately, but exactly 610 genes that were expressed. From these 16 transcripts, RNA transcripts that were obtained from the different sample of cells they looked at. And they took those genes that were containing those transcripts and mapped them out here. You got a phylogenetic tree with different organisms that share those genes in this heat map on the right. Excuse me. You can see muscle up here at the top, and how they put it on a scatter plot and compared it to non-magnetic and other magnetic cells. And so it's important to see that on this scatter plot and this logarithmic configuration they did, that down here you've got this cluster, which is another piece of supporting evidence that these genes are conserved evolutionary-wise. And so it's significant that these two different clusters showed up from the same cellular source on this left side. I really like this figure on the right in that it's a heat map, and the more intense the red, the more those genes were shared by different organisms, or the more they were shared by an increasing amount of organisms. And you can see that, muscle showed up good as their control, and then the non-magnetic and the magnetic are in this sort of alternating sequence of light blue, dark blue, light blue, dark blue. Talking about results, they saw 11 genes that were homologous with other eukaryotes, and 9 of those genes contained genes that were shared with archaea. So all three domains are right here included. And then they went a little further and compared genes that are shared between eukaryotes. And if you look at figure 2a here, you'll see this purple bluish figure. The darker purple it is means more genes were shared between eukaryotes, and the X's indicate that no genes were shared. And if you look at the numbers to the left of this picture in 2a, you can see the actual number of genes that were shared. If you can refer to page 3, bottom right, you can see the author states that these core genes, along with MAMH, MAMN, 6 of those ones that were shared between bacteria and eukaryotes, they were all part of the same operon that is responsible for producing magnetite crystallization, apart from one little caveat. To me, that's definitive. And I think this kind of just puts the nail in their coffin that these cells are not just a fluke in trout, and it's a real mechanism that's seen in eukaryotes that's conserved evolutionary. We can trace it back to the ancestor. And so if we find these cells or these genes in birds or another organism we're trying to discern on the nature of their geomagnetism navigation, it's just going to strengthen that evidence. So second to last figures 2b through g, I want to look at these scatter plots at the top right. They aligned all these sequences, and then you can see in B, 2b here, that's all the taxa combined with other taxa and noise, background noise in gray. They're clustered down here based on the alignment in the sequence in H, which is another supporting step in their claim that these are conserved genes. And so finally, this last table, if you look, it is consisting of homologous and paralogs of zebrafish and salmon compared to the 11 bacterial genes that they identified that produce magnetite and are conserved in eukaryotes. So the first column gives the name of the protein, and if you go over to the fourth, you can see within eukaryotes some of the jobs of these proteins. I see stuff like microtubule binding, protein couple receptors, tight junctions, a lot of protein involved in, or a lot of proteins involved in cellular adhesion. It's just neat to look at and see, you know, all these proteins are generally related. They're from the same operon, and because they're so close on the conserved sequence that they looked at, that is just red hot evidence, in my opinion, that these are evolutionarily related. To conclude, it's honestly kind of nice to see a paper that I can walk away from and be like, yeah, they did what they said they set out to do. There's still a lot of untangling how animals certainly navigate in terms of a magnetic sense. We can for sure say animals are affected by a geomagnetic field or a field that's made in a lab. And what is most supporting from this paper for me is that, one, they discerned that magnetite is produced within the cells of the trout and not from some alternative source. They, in my opinion, definitively said, okay, these cells have genes that will allow for that production, and even though they're right beside other cells that have similar function, they don't have those genes turned on. And then their genetic comparison and data that they obtained of conserved sequences and how these are related between bacteria, archaea, and eukaryotes today, I don't see how it really could be refuted, honestly. I'm sure there is a way, because I am not a geneticist by any means. But overall, I hope you all enjoyed this presentation. I hope it wasn't too tedious for you. Thank Dr. Ashley and my classmates for allowing me to just be part of it, kind of on the sidelines online. And I wish you all the best for the rest of your school and the rest of the semester. Have a great evening. If you were wondering about that little asterisk at the beginning of the slideshow next to the word dogs, it's because of this paper that I came across in getting this presentation together. Apparently dogs, whenever they defecate, they will align their bodies with north and south magnetic poles. So just be Be sure to remember that important information. Thank you.